Hey guys, welcome to another episode of the Human AI podcast. Today we're talking about formation of urine formation. So I've got Grog here with me, so it can help us out. Grog, tell us, how do we form urine and why is it important?
Yo, thanks for the intro. All right, let's break down urine formation. It happens in the kidneys, which are like your body's filtration champs. The kidneys filter blood to remove waste, excess water, and electrolytes, turning it into urine. This keeps your body balanced, no buildup of toxins, stable blood pressure, and proper electrolyte levels. The process has three main steps: filtration, reabsorption, and secretion. First, blood gets filtered in the nephrons, tiny kidney units, where water, glucose, salts, and waste pass into a filtrate. Second, the body reabsorbs what it needs, like most of the water, glucose, and sodium, back into the blood. Third, the kidneys add extra waste or ions, like potassium or drugs, into the filtrate. That final mix becomes urine, flows to the bladder, and you
So,
pee it.
so the secretion part is explain more. So it's basically things that haven't been filtered that are gonna get peed through another way.
Exactly, you nailed the idea. Secretion and urine formation is when the kidneys actively add stuff to the filtrate that wasn't caught during the initial filtration. After the nephrons filter blood, secretion happens in the tubules, where cells in the kidney pump out extra waste, drugs, or ions, like potassium, hydrogen, or ammonia, into the filtrate to be excreted as urine. It's like the kidneys saying, "We need to get rid of this too." This step fine tunes blood chemistry and clears out things that filtration missed,
Hmm.
ensuring toxins or excess substances don't stick around. Want me
And
to
what about the anatomical location of the kidneys from an evolutionary perspective? Why did they end up in that location? How does their location kind of correlate to their function?
Great question. The kidneys are located in the retroperitoneal space, tucked against the back of the abdominal cavity, just below the rib cage, on either side of the spine, roughly at the level of the T12 to L3 vertebrae. From an evolutionary perspective, this spot makes sense for a few reasons. First, being deep in the body and shielded by the rib cage and muscles offers protection for these vital organs, which is crucial since they filter blood and maintain homeostasis. Early vertebrates, like fish, had kidneys along their body length, but as tetrapods evolved to live on land, kidneys shifted to a more compact, centralized location to efficiently handle blood flow from a larger circulatory system. Their position near the spine ties to their reliance on major blood vessels. The renal arteries branch directly off the aorta, and the renal veins feed into the inferior vena cava, giving them quick access to the body's blood supply for filtration. This spot also keeps them close to the adrenal glands, which sit on top and release hormones like aldosterone that regulate kidney function. The retroperitoneal placement minimizes movement during activity, stabilizing blood flow to the nephrons. So, their location is a balance of protection, vascular access, and hormonal coordination, all critical for their role in filtering blood, balancing fluids, and excreting waste.
Yeah, but I don't
Want me
understand why don't we have them higher? Huh?
Good question. Why not higher up, like in the chest or something? The kidney's position in the retroperitoneal space, just below the rib cage, comes down to evolutionary trade offs. Placing them higher, say in the thorax, would crowd vital organs like the lungs and heart, which need space to expand and contract for breathing and pumping. The kidneys need a stable, high pressure blood supply from the aorta, and their current spot near the spine taps directly into that major arterial highway without competing with the heart's plumbing.
Yeah, but why are the lungs where they are? I'm just wondering, is there like in the subject of evolutionary medicine, is there a book that talks about why different organs are in different locations?
You're diving deep into evolutionary medicine, and I love it. The lungs are in the thorax, high in the chest, because they need maximum space to expand for efficient gas exchange. And being close to the heart ensures quick oxygen delivery to the blood. Their placement also ties to early vertebrate evolution, where gills, lung precursors, were near the mouth and heart for water flow, and lungs later evolved in a similar anterior position for air. As for your question about a book, there's no single definitive book just on why organs are located where they are. But Why We Get Sick by Randolph Nesse and George C. Williams touches on evolutionary medicine and includes insights on organ placement as trade offs for survival and function. Another good one is The Story of the Human Body by Daniel Lieberman, which explores how evolution shaped anatomy, including why organs like kidneys or lungs ended up where they did.
Yeah, this
These
question, evolution of, yes, how evolution shaped anatomy and history of anatomy, it's a subject I'm very interested. Give me the best few books for this.
Awesome. Glad you're pumped about how evolution shaped anatomy. Here are three top books that dive into this subject, blending evolutionary biology with anatomical history. 1. The Story of the Human Body by Daniel Lieberman. This is a fantastic read that walks through how evolutionary pressures shaped human anatomy, from why our organs are where they are to how bipedalism changed our structure. It's clear, engaging, and ties directly to why our bodies look the way they do. 2. Why We Get Sick by Randolph Nesse and George C. Williams, a classic in evolutionary medicine. This book explains how natural selection influenced organ placement and function, including why some anatomical designs are trade offs that leave us prone to certain diseases. It's a bit broader, but super relevant. 3. Your Inner Fish by Neil Shubin. This one traces the evolutionary history of human anatomy back to our fish ancestors, showing how structures like lungs and kidneys evolved and why they're positioned as they are. Shubin's storytelling makes it a fun yet deep dive. These books give you a solid mix of evolutionary theory, anatomical insights, and historical context. Want me to search for more or narrow it down to one?
Well, no, these three are great. I'm gonna check these. Is there one that focuses on systems biology or whatever, systems medicine or whatever it's called, and evolutionary medicine?
Nice. Glad those books hit the spot. For systems biology or systems medicine combined with evolutionary medicine, the pickings are a bit slimmer, but there's one standout that bridges these fields. Evolutionary Systems Biology by Orkin S. Sawyer, editor. It's a collection of essays diving into how evolutionary principles shape biological systems, including how networks of genes, proteins, and organs, like the kidney or respiratory system, evolve to work together. It's a bit technical, but connects systems biology, looking at the body as interconnected networks, with evolutionary trade offs, like why certain anatomical setups persist despite flaws. Another option.
Interesting. Tell me, sorry, you said another option, what?
My bad for cutting off there. Another option is The Logic of Life by Francois Jacob. It's a classic that leans into how evolution shapes biological systems, touching on how organs and their functions emerged as interconnected networks. Though it's less technical than Sawyer's book. It blends evolutionary medicine with a systems like perspective on why our anatomy works the way it does.
Okay, let's go back to our topic. So the glomerulus filtrates the blood, then the proximal tubule reabsorbs from the nephron. And do we have secretion into the nephron too in the proximal tubule? And if yes, why specifically at the proximal tubule? And so tell me also, in the loop of Henle, what's going on? In the distal tubule, do we modify the fluid? What's going on there? And then in the collecting duct?
All right, let's jump back into urine formation and break down what's happening in each part of the nephron. Starting with your question about the proximal tubule and secretion. Yes, secretion does happen in the proximal tubule. It's where the kidneys actively pump substances like drugs, toxins, and excess ions, like potassium or hydrogen, from the blood into the filtrate to be excreted. Why here? The proximal tubule is loaded with transport proteins and has a huge surface area, making it super efficient for both reabsorption, taking back water, glucose, and sodium, and secretion, dumping waste. It's like the nephron's multitasker, handling both at once to fine tune what stays or goes. Now, the loop of Henle. This is where the kidney creates a concentration gradient in the medulla to control water and salt balance. The descending limb is permeable to water, so water gets reabsorbed as the filtrate moves into the salty medulla, concentrating the fluid. The ascending limb is impermeable to water, but pumps out sodium, chloride, and potassium, diluting the filtrate. This setup allows the kidney to decide how concentrated the urine will be later. In the distal tubule, the filtrate is modified further. It's less about bulk reabsorption and more about fine tuning. The distal tubule reabsorbs sodium and calcium and secretes more potassium or hydrogen ions, depending on what the body needs. Hormones like aldosterone kick in here to regulate sodium and potassium balance, tweaking the filtrate's composition. Finally, the collecting duct is the last stop. It's where the urine's final concentration is set. Antidiuretic hormone, ADH, makes the collecting duct more permeable
Oh
to
god,
water.
dude, you're talking good stuff, but it's so much. Maybe kind of summarize in a sentence or two.
All right, here's the quick version. The proximal tubule reabsorbs essentials like water and glucose and secretes wastes like drugs due to its transport heavy setup. The loop of Henle creates a salt gradient to control urine concentration. The distal tubule fine tunes sodium and potassium, and the collecting duct adjusts water reabsorption to
All right.
finalize urine
So,
concentration.
so yeah, but I'm just wondering, so secretion happens only in the proximal tubule.
Actually, secretion doesn't happen only in the proximal tubule, but it's a major player there. The proximal tubule is the primary site for secretion, where cells use active transport to dump drugs, toxins, and ions, like potassium and hydrogen, from the blood into the filtrate, thanks to its rich supply of transport proteins.
Hmm.
How secretion also occurs in the distal tubule and the collecting duct. In the distal tubule, cells secrete potassium and hydrogen ions to regulate blood pH and electrolyte balance, often driven by hormones like aldosterone. The collecting duct can also secrete hydrogen ions or bicarbonate to fine tune acid base balance, though its main role is water reabsorption. These later segments focus on specific ions rather than the broad waste dump of the proximal tubule.
So,
Want me?
so wait a sec. So, with why the loop of Henle doesn't do secretion, and also if I really like asking a lot of whys, what part of medicine will I really enjoy? One answer is evolutionary medicine, but maybe evolution is not the only answer for the why.
All right, let's tackle both questions. First, why doesn't the loop of Henle do secretion? The loop of Henle is specialized for creating a concentration gradient in the kidney's medulla to regulate water and salt balance, not for secretion. Its descending limb passively reabsorbs water, and the ascending limb actively pumps out sodium, chloride, and potassium to dilute the filtrate, setting up the osmotic environment for urine concentration later. It lacks the active transport mechanisms and enzymes needed for secretion, which are concentrated in the proximal tubule, distal tubule, and collecting duct, where blood to filtrate transfer of ions or drugs happens. The loop's job is more about reabsorption and gradient building, not adding stuff to the filtrate. Now, for your love of asking why in medicine besides evolutionary medicine, you'd likely thrive in pathophysiology or medical research. Pathophysiology is all about digging into the why of disease processes, why symptoms arise, why organs fail, and how systems go wrong, like why a specific ion imbalance causes a symptom. It's detective work at the cellular and systemic level. Medical research, especially in fields like molecular biology or clinical investigation, also feeds that curiosity, letting you explore why certain treatments work or
Interesting.
why dis-
Okay, so what in the structure or location of loop of Henle, or maybe some other quality of loop of Henle, makes it not good for secretion.
The loop of Henle's structure and function are tailored for reabsorption and creating a concentration gradient, not secretion. Its thin descending limb is highly permeable to water, but lacks the active transport proteins needed to move substances like ions or drugs from blood to filtrate. The thick ascending limb actively pumps sodium, chloride, and potassium out of the filtrate into the surrounding medulla, but doesn't have the machinery, like organic anion transporters found in the proximal tubule, for secreting wastes or drugs.
And why does it lack these? Huh? Why does it lack these transporters or whatever?
I think you meant passport carousel as a
No,
mix up.
dude, I said
Maybe
why.
referring.
No, no, I said why does it lack these transporters?
My bad, I misheard you there. All right, why does the loop of Henle lack secretion transporters? The loop of Henle is evolutionarily specialized for creating a concentration gradient to regulate urine concentration, not for secretion. Its cells, especially in the descending and ascending limbs, are packed with aquaporins for water reabsorption and ion pumps like the NaKCl co-transporter for moving salts out of the filtrate. It lacks secretion specific transporters like organic anion or cation transporters because its role is to set up the osmotic environment in the medulla, not to actively dump drugs or ions into the filtrate. Those
Interesting,
secretion
interesting.
transporters are concentrated in the proximal tubule, where the blood supply and cellular machinery are optimized for filtering and secreting a wide range of substances.
Okay,
The
that makes more sense. All right, let's continue and let's talk a bit. Let's talk a bit about the glomerulus. Specifically, let's talk about how filtration is dependent on like blood pressure and yeah, the difference in diameter between afferent and efferent.
Cool. Let's zoom in on the glomerulus and how filtration ties to blood pressure and vessel diameter. The glomerulus is a network of capillaries in the nephron where blood filtration starts. Blood pressure drives filtration by pushing plasma through the glomerular capillaries into Bowman's capsule, forming the filtrate, water, ions, glucose, and waste, but not big proteins or cells. This process, called glomerular filtration, depends heavily on the hydrostatic pressure in the capillaries, which is directly influenced by systemic blood pressure. If blood pressure drops too low, filtration slows. If it's too high, it can strain the glomerulus. The difference in diameter between the afferent and efferent arterioles is key. The afferent arteriole, which brings blood into the glomerulus, is wider, allowing high pressure blood to flow in easily. The efferent arteriole, which carries blood out, is narrower, creating a bottleneck that maintains high pressure in the glomerular capillaries, pushing filtrate out. This pressure
So
gradient.
wait, let me ask you. So what about these other things like we have podocytes and we have these pores. Can you explain it in regard to these terms too?
All right, let's dive into how podocytes and pores fit into glomerular filtration, tying it to blood pressure and arterial dynamics. The glomerulus filters blood through a specialized barrier, and podocytes are key players. These are star shaped cells that wrap around the glomerular capillaries, forming a filtration barrier with tiny gaps called slit diaphragms. The pores you mentioned are part of this setup, specifically the fenestrae, which are holes in the capillary endothelium, and the slit diaphragms between podocyte foot processes. Together, they act like a sieve, letting water, ions, glucose, and small waste molecules pass into Bowman's capsule
But in just a
while blocking
sentence
what.
or in just a sentence or two, why do we have both podocytes and we have also the pores? Isn't one enough?
Podocytes and pores, fenestrae and slit diaphragms, work together because the pores allow small molecules to pass quickly, while podocytes' slit diaphragms add a finer filter to block larger proteins, ensuring precise filtration. One alone wouldn't be selective enough to balance speed and accuracy.
Interesting. Sorry, sir, I was speak. What are you talking about? We were speaking in English a minute ago. Anyway, having said that, so podocytes are like epithelial cells. They're attached to the membrane, basically. And what are these pedicels?
No worries. Let's stay in English and get back to podocytes. You're right. Podocytes are specialized epithelial cells in the glomerulus, attached to the basement membrane of the glomerular capillaries. Their job is to form a filtration barrier. The pedicles, or more accurately, foot processes, are like tiny finger-like extensions of the podocytes that interlock around the capillaries, creating narrow gaps called slit diaphragms. These foot processes are critical because they act as a final filter, letting small stuff like water and ions pass into the filtrate while blocking bigger molecules like proteins. Think of them as the podocyte's fingers gripping the capillary, with just enough space between them to be selective. Want me to clarify anything else about this setup?
I don't understand the difference between them. Also, why would like glucose in diabetes damage the podocytes? Huh?
All right, let's clear
Tell me.
up the difference between podocytes and their. I think you might mean pedicles or foot processes when you say pedicels.
Yeah.
But all right, got it. Podocytes are specialized epithelial cells in the glomerulus that wrap around capillaries to form a filtration barrier. Their foot processes, often called pedicles, are finger-like extensions that interlock, creating slit diaphragms that filter blood while blocking large proteins.
I don't understand the difference. Dude, bro.
Want to keep
So
going
what's
with
so what? No, tell me what's the difference between pedicels and podocytes?
All right, let's nail this down. Podocytes are the actual cells in the glomerulus, star-shaped epithelial cells that form the filtration barrier around capillaries. Pedicels, or more commonly called foot processes, are the tiny, finger-like extensions of podocytes that interlock with each other, creating the slit diaphragms that act like a sieve to filter blood while keeping out big molecules like proteins. All right, let's make it crystal clear. Podocytes are the cells in the glomerulus that sit on the outside of the capillaries, acting like the main structure of the filtration system. Their foot processes, or pedicels, are like little arms extending from these cells that weave together, creating a net with tiny gaps, slit diaphragms, that filters blood, allowing small molecules like water and salts through, but stopping larger ones like proteins. Think of podocytes as the cell body, and pedicels as their outstretched fingers doing the filtering work together. Does
And
that
so
help?
okay, so the pedicels are the small cells and the podocytes the big cells. And then the podocytes, after the podocytes, we go through the glomerulus.
Okay, let's straighten this out, because I think there's a bit of a mix up. Pedicels or more accurately, foot processes, aren't small cells. They're extensions of podocytes. Podocytes are the cells, not big cells, but specialized glomerulus cells that wrap around capillaries. These podocytes have finger-like projections called foot processes, or pedicels, that interlock to form the filtration slits. There aren't two separate cell types here. It's one cell, the podocyte, with its foot processes doing the filtering job. Now, about the
Dude, I am
glomerulus.
confused.
No worries, let's simplify this. Podocytes are cells in the glomerulus that help filter blood. Their foot processes, sometimes called pedicels, are like tiny fingers sticking out from these cells that interlock to form a filter, letting small stuff like water through but blocking big stuff like proteins. There's no separate small or big cell, just podocytes and their extensions working together in the glomerulus, which is the capillary network where filtration happens. Want me to break it down another way or move to the next step after filtration?
It's not getting through. Maybe if you could explain it another way.
All right, let's try a different angle to make this click. Imagine the glomerulus as a coffee filter in your kitchen. The podocytes are like the paper filter itself, a single layer of material, the cells, wrapped around the coffee grounds, the capillaries. The foot processes, or pedicels, are like the tiny holes in that filter paper, created by the podocytes' finger-like extensions interlocking tightly. These holes let the coffee, small molecules like water and ions, drip through into the pot, Bowman's capsule, but keep the grounds, big proteins, out. It's all one unit. Podocytes are the filter, and their foot processes are the holes doing the filtering in the glomerulus.
Okay, got it. So let's talk a bit about the autoregulation of the glomerular filtration rate.
Nice, glad that clicked. All right, let's dive into autoregulation of the glomerular filtration rate, GFR. The kidneys keep GFR, the rate at which blood is filtered in the glomerulus, steady despite changes in blood pressure, thanks to autoregulation. This happens mainly through two mechanisms, the myogenic reflex and tubuloglomerular feedback. The myogenic reflex is when the afferent arteriole, the vessel bringing blood to the glomerulus, senses high blood pressure
Can you
and
talk? Sorry for interrupting. Can you talk a bit faster? Because I'm gonna fall asleep, dude.
Haha, my bad. Let's speed this up. Kidneys autoregulate GFR to keep filtration steady no matter blood pressure swings. Myogenic reflex. Afferent arteriole senses high pressure, tightens to limit blood flow. Low pressure, it relaxes. Tubuloglomerular feedback. Macula densa cells in the distal tubules sense fast filtrate flow, signal the arteriole to constrict, slowing GFR. Keeps filtration balanced, protects glomerulus.
So it's
Want more?
basically through vasoconstriction and vasodilation through the sympathetic and parasympathetic.
Not quite. Autoregulation of GFR isn't primarily through the sympathetic nervous system, though that can play a role in bigger stress responses. It's mainly driven by local mechanisms in the kidney itself. The myogenic reflex causes vasoconstriction or vasodilation of the afferent arteriole based on blood pressure changes. Stretch from high pressure makes the arteriole constrict. Low pressure lets it dilate, all without nerve input. Tubuloglomerular feedback involves the macula densa sensing sodium chloride levels in the filtrate. High flow triggers vasoconstriction of the afferent arteriole via local signals, not sympathetic nerves. Sympathetic nerves might kick in during extreme cases, like shock, to override and reduce GFR. But day-to-day autoregulation is the kidney's own show. Want to clarify anything here?
I'm confused.
No worries, let's simplify. Autoregulation of GFR keeps kidney filtration steady using two main tricks, and they're not controlled by sympathetic nerves. First, the myogenic reflex. If blood pressure is high, The afferent arteriole automatically tightens to slow blood flow. If low, it widens. Second, tubuloglomerular feedback. Special cells sense too much filtrate flow and tell the arteriole to constrict, slowing things down. These are local kidney mechanisms, like an internal thermostat, not nerve-driven. Does that clear it up?
Kind of.
Or one?
How does ANP, nitric oxide, prostaglandins, dopamine, kinins, how do these play a role in all of this? Also, angiotensin 2, noradrenaline. Yeah.
All right, let's zoom through how these players, angiotensin 2, nitric oxide, prostaglandins, dopamine, kinins, and noradrenaline, tie into autoregulation of glomerular filtration rate, GFR. Angiotensin 2 constricts both afferent and efferent arterioles, but especially the efferent, boosting glomerular pressure to maintain GFR when blood pressure drops. Nitric oxide and prostaglandins are vasodilators, relaxing the afferent arteriole to increase blood flow, and GFR if pressure's too low, countering constriction. Dopamine, at low doses, dilates renal arteries, enhancing blood flow to the glomerulus to support GFR. Kinins, like bradykinin, also promote vasodilation, helping maintain glomerular blood flow. Noradrenaline, tied to sympathetic nerves, constricts arterioles during stress, like shock, reducing GFR to prioritize other organs, but it's not a big player in daily autoregulation. These molecules fine-tune the kidney's local myogenic and tubuloglomerular mechanisms to keep GFR stable. Want me to focus on one of these or move on?
What about things like adenosine and alpha 2 receptors, alpha 2A, alpha 2B receptors?
All right, let's tackle adenosine and alpha 2 receptors, like alpha 2A and alpha 2B, in the context of GFR autoregulation. Adenosine plays a big role in tubuloglomerular feedback. When the macula densa in the distal tubule senses high sodium chloride in the filtrate, meaning fast filtration, it releases adenosine, which binds to receptors on the afferent arteriole, causing vasoconstriction. This slows blood flow to the glomerulus, reducing GFR to prevent overload. Alpha 2 receptors, including subtypes like alpha 2A and alpha 2B, are part of the sympathetic response, not the kidney's core autoregulation. When activated by catecholamines like noradrenaline, they can mildly constrict renal arterioles, reducing GFR during systemic stress, EGG, low blood pressure, or shock. But their role is secondary to local mechanisms like the myogenic reflex or adenosine-driven feedback. Adenosine's the star for fine-tuning GFR locally, while alpha 2 receptors are more about broader stress responses. Need more on this or something else?
I need more on this.
All right, let's dig deeper into adenosine and alpha 2 receptors in GFR autoregulation. Adenosine is a key player in tubuloglomerular feedback. When the macula densa detects high sodium chloride in the filtrate, indicating rapid filtration, it boosts adenosine release. Adenosine then acts on A1 receptors on the afferent arteriole, triggering vasoconstriction, which reduces blood flow into the glomerulus and lowers GFR to keep filtration balanced. It's like a brake pedal to prevent the kidneys from overworking. In contrast, alpha 2 receptors, alpha 2A and alpha 2B, are more about systemic control. They're found on renal arterioles and respond to noradrenaline or epinephrine from sympathetic nerve activation or stress. When stimulated, they cause mild vasoconstriction, which can decrease GFR, but this usually happens in extreme cases like hemorrhage or dehydration.
Okay.
Not every day.
So, what about the renal blood flow? How does that auto-regulate?
Renal blood flow, RBF, autoregulation, keeps the kidney's blood supply steady despite changes in systemic blood pressure, ensuring consistent filtration and function. It works through mechanisms similar to GFR autoregulation, mainly the myogenic reflex and tubuloglomerular feedback. In the myogenic reflex, the afferent arteriole senses high blood pressure via stretch and constricts to limit blood flow, or dilates when pressure drops, maintaining stable RBF. Tubuloglomerular feedback involves the macula densa, sensing sodium chloride levels in the filtrate. High levels, indicating too much flow, trigger adenosine release, which constricts the afferent arteriole, reducing RBF.
Can you keep your answers limited to two sentences when we speak?
Sure thing. Renal blood flow is kept steady by the myogenic reflex, where the afferent arteriole constricts or dilates based on blood pressure changes, and tubuloglomerular feedback, where high filtrate flow triggers adenosine to constrict the arteriole. Nitric oxide and prostaglandins fine-tune this by promoting vasodilation when needed.
So, what makes prostaglandins special to do this, and nitric oxide, and ANP, and kinins to dilate, and whatever?
Prostaglandins and nitric oxide are special because they act as local vasodilators, relaxing the afferent arteriole to boost renal blood flow when blood pressure drops. Angiotensin II, kinines, and other vasodilators like dopamine counteract constriction, ensuring stable flow and protecting kidney function.
Yeah, but you didn't tell me how they're special. What in the structure makes the. How does the structure relate to the function? What in the structure allows them to have this function?
Prostaglandins and nitric oxide are small, lipid-soluble molecules that rapidly diffuse to smooth muscle cells in the afferent arteriole, triggering relaxation by activating pathways like CAMP or CGMP. Their simple structure allows quick synthesis and action, unlike larger molecules, ensuring fast vasodilation to maintain renal blood flow.
Okay, let's put a pause on this just for a bit, and let's come to another topic. We'll come back to this. When it comes to glomerular filtration, what does ultrafiltration mean?
Ultrafiltration in glomerular filtration refers to the process where blood plasma is filtered under high pressure through the glomerular capillaries, fenestra, and podocyte slit diaphragms. It produces a nearly protein-free filtrate containing water, ions, glucose, and small wastes, driven by hydrostatic pressure.
I don't understand the difference between filtration and ultrafiltration.
Filtration is the general process of separating substances, like blood plasma passing through the glomerulus to form filtrate. Ultrafiltration specifically describes this glomerular process where high pressure forces a nearly protein-free filtrate through the fine fenestra and podocytes slit diaphragms.
What's the difference?
Filtration is the broad term for any process where substances are separated, like straining coffee. Ultrafiltration is a specific, high-pressure type of filtration in the glomerulus, where only small molecules like water and ions pass
Okay,
through the.
fair enough. Uh, and why do we call it
Cool.
tubular fluid?
Tubular fluid is the name for the liquid in the nephron tubules, after ultrafiltration in the glomerulus, as it flows through structures like the proximal tubule and loop of Henle. It's called tubular fluid because its composition changes from the initial filtrate due to reabsorption and secretion in the tubules.
Hmm. Okay, interesting. And so let's talk a bit about the reabsorption from the proximal tubule. So we said it becomes the tubular fluid at this point, and you have to explain to me again why. And we have filtered water, sodium, potassium, chloride, bicarbonate, and all of this is absorbed back to the blood.
The liquid post-glomerular filtration is called tubular fluid because it enters the nephron's tubules, where its composition shifts due to reabsorption and secretion, distinguishing it from the initial filtrate. In the proximal tubule, about 65 to 70% of filtered water, sodium, potassium, chloride, and bicarbonate is reabsorbed back into the blood via active transport, like sodium pumps, and passive diffusion to maintain body fluid and electrolyte balance.
Hmm. And how does the sodium potassium ATPase play a role here?
The sodium potassium ATPase pump in the proximal tubule actively transports sodium out of tubule cells into the blood, creating a low sodium gradient inside the cell. This gradient drives sodium reabsorption from the tubular fluid, pulling along water, chloride, and other solutes via co-transporters and osmosis.
What about transcellular and paracellular reabsorption?
Paracellular reabsorption in the proximal tubule happens when water, chloride, and small ions like sodium pass between tubule cells through tight junctions, driven by osmotic and concentration gradients. Transcellular reabsorption, in contrast, involves substances moving through the cells, using transporters like the sodium potassium ATPase to actively reabsorb sodium, glucose, and others.
So, what I don't get it.
Paracellular reabsorption is when stuff like water and chloride slips between proximal tubule cells through leaky tight junctions, pulled by osmosis or concentration differences. Transcellular reabsorption is when substances like sodium or glucose move through the cells, using pumps like sodium potassium ATPase or specific transporters.
I still don't get the difference.
All right, let's make it super clear. Paracellular reabsorption means water, chloride, or ions move between the tubule cells, squeezing through gaps, tight junctions, like sneaking through a fence, driven by osmosis or gradients. Transcellular reabsorption means substances like sodium or glucose go through the cells, crossing cell membranes using
Okay,
pumps.
got it. Got it. Okay. So this sodium potassium ATPase, so it pumps out sodium, and then this is along a chemical. And so how does this happen? It's in regard to chemical and electrical gradients.
The sodium potassium ATPase pump in the proximal tubule pumps sodium out of tubule cells into the blood, creating a low sodium concentration inside the cell and a slight electrical gradient. Cell interior becomes negative. This chemical, low sodium, and electrical, negative charge, gradient drives sodium from the tubular fluid into the cell via transporters, pulling along water and other solutes like glucose or chloride.
So it's along a chemical and against an electrical, but I don't get why.
The sodium potassium ATPase pump moves sodium out of the tubule cell to the blood, making the cell's interior low in sodium and slightly negative. This creates a chemical gradient. Sodium wants to flow into the cell, and an electrical pull. Positive sodium is attracted to the negative cell, driving sodium reabsorption from the tubular fluid.
Okay, let's go back to the autoregulation of glomerular filtration rate. So why do prostaglandins ANP and dopamine and nitric oxide and kinase, why do they dilate and not constrict?
Prostaglandins, nitric oxide, dopamine, and kinins dilate the afferent arteriole because they activate signaling pathways like CAMP or CGMP that relax smooth muscle cells, increasing blood flow to maintain GFR when pressure drops. Their molecular structure and receptor interactions favor vasodilation over constriction, unlike angiotensin II, which binds receptors that trigger muscle contraction.
So in this renal response to low blood pressure, what does the distal tubule do? What does the macula densa do? What do the juxtaglomerular cells do? And why?
When blood pressure drops, the macula densa in the distal tubule senses lower sodium chloride in the filtrate and signals juxtaglomerular cells to release renin, kickstarting the renin-angiotensin-aldosterone system.
But why are
RAAS.
these cells the ones that are in charge for these roles, like the macula densa and the juxtaglomerular?
The macula densa and juxtaglomerular cells are specialized for these roles due to their strategic location and unique structure at the juxtaglomerular apparatus, where the distal tubule meets the afferent arteriole. Macula densa cells have sensitive ion channels to detect sodium chloride changes, signaling juxtaglomerular cells, which are packed with renin-filled granules, to release renin and regulate blood pressure via RAAS.
Okay, let's go back to the sodium reabsorption now from the proximal tubule, and let's talk a bit more. Actually, before we go there, let's talk a bit more about the sodium potassium ATPase, and again about the difference between a chemical and electrical gradient in a sentence.
The sodium-potassium ATPase pump creates a chemical gradient by pumping sodium out of proximal tubule cells, making the cell interior low in sodium, and an electrical gradient by making the cell interior negative, both driving sodium reabsorption from the tubular fluid. The chemical gradient pulls sodium into the cell due to its concentration difference, while the electrical gradient attracts positively charged sodium to the negatively charged cell interior.
Yeah, but I don't get why they're called chemical and electrical.
The chemical gradient refers to the difference in sodium concentration, more outside the cell, less inside, created by the sodium-potassium ATPase, driving sodium movement into the cell. The electrical gradient is the voltage difference, cell interior negative, outside positive, that pulls positively charged sodium into the negatively charged cell.
I don't get it.
All right, let's break it super simple. The chemical gradient is like a hill of sodium, high outside the cell, low inside, so sodium rolls into the cell to balance it. The electrical gradient is like a magnet, where the cell's negative charge inside pulls the positive sodium in, both set up by the sodium-potassium ATPase pump.
I don't get it.
Okay, let's try a different angle. The sodium-potassium ATPase pump makes two forces: a chemical gradient, where sodium wants to move into the cell because there's less sodium inside, like water flowing to a dry spot, and an electrical gradient, where the cell's negative charge inside acts like a magnet, pulling positive sodium in. Both help suck sodium from the tubular fluid back into the cell. Want me to try another analogy?
Yeah. And use an analogy to explain this along and against.
Imagine a river, tubular fluid, with fish, sodium, wanting to swim into a pond, the cell. The chemical gradient is like a current pushing fish into the pond because the pond has fewer fish, low sodium inside, driving sodium along this concentration flow. The electrical gradient is like a net pulling fish in because the pond is negatively charged, attracting positive sodium against other forces.
I don't get it. I don't even get why their name relates to their function.
All right, let's simplify and explain why chemical and electrical gradients are named that way and how they work. The sodium potassium ATPase pump moves sodium out of the cell, creating two driving forces for sodium reabsorption. The chemical gradient is called chemical because it's about the concentration of a substance, sodium. There's more sodium outside the cell than inside, so sodium flows into the cell like water moves from a full bucket to an empty one. The electrical gradient is called electrical because it's about charge. The cell's interior is negatively charged, so it pulls in positively charged sodium, like a magnet attracts metal. Both gradients, set up by the pump, make sodium move from the tubular fluid into the cell for reabsorption. Does this analogy or explanation help, or want me to try another way?
Try another way.
Okay, let's picture a candy store, the cell, and a kid with candies, sodium, outside. The chemical gradient is called that because it's about the amount of candy. Fewer candies inside the store than outside. So candies, sodium, rush in to fill the gap, like kids running to a less crowded store. The electrical gradient is called electrical because the store has a negative vibe, negative charge, pulling in positive candy, sodium, like a cool party vibe draws kids. Both driven by the sodium potassium ATPase, pumping candies out.
Yeah, but
Want me
why
to
is
tweak?
one along and the other against?
I think there might be some confusion about along and against
Yeah,
from the earth.
because sodium, potassium, ATPase, we have this is along the chemical and against the electrical gradients. The pumping out of sodium from cells into the blood.
Got it. Thanks for clarifying. The sodium potassium ATPase pumps sodium out of the proximal tubule cell into the blood along the chemical gradient because sodium moves from a high concentration inside the cell to a lower concentration outside, like rolling downhill. It pumps sodium against the electrical gradient because the cell's interior is negative, wanting to pull positive sodium back in. So the pump has to push sodium out despite this electrical pull, like swimming upstream.
What even is, dude? I still don't understand electrical and chemical and all of this. Where do they get the name from?
All right, let's zero in on why we call them chemical and electrical gradients and clear this up. The sodium potassium ATPase moves sodium out of the proximal tubule cell into the blood, setting up two forces for sodium movement. The chemical gradient gets its name because it's about the concentration of a chemical, sodium, high inside the cell, low outside. So sodium naturally flows out to balance it, like sugar dissolving from a dense pile into water. The electrical gradient is named electrical because it's about electric charge. The cell's negative charge inside wants to hold on to positive sodium, but the pump forces sodium out against this pole, like pushing a magnet away from an opposite charge. These names reflect what drives sodium: concentration for chemical, charge for electrical. Does this help or want another analogy?
Um, so chemical is because of the chemical, electrical is because of.
Exactly. Chemical gradient is named because it's driven by the concentration of a chemical, sodium, with sodium moving from high to low concentration, like water flowing to a less salty area. Electrical gradient is named because it's driven by electric charge, where the cell's negative charge pulls positive sodium, but the sodium potassium ATPase pushes it out against this charge, like repelling magnets.
All right. Um, let's talk a bit about facilitated diffusion then. Uh, of chloride. And let's talk about what's going on with phosphate and sulfate and all of these when with sodium reabsorption. How do they connect?
In the proximal tubule, facilitated diffusion of chloride occurs via paracellular pathways, where chloride follows sodium's reabsorption, driven by sodium potassium ATPase due to electrical and concentration gradients, like tagging along with a crowd. Phosphate, sulfate, and other anions are reabsorbed transcellularly through sodium-coupled cotransporters, piggybacking on sodium's movement into the cell to maintain electrolyte balance.
Okay. And what about biocarbonate when it comes to water reabsorption? What about what's going on with biocarbonate and chloride and sodium?
Bicarbonate is reabsorbed in the proximal tubule via sodium-coupled mechanisms, where sodium's movement, driven by sodium-potassium ATPase, helps convert bicarbonate into CO2 and water inside tubule cells, then reabsorbs it into the blood to regulate pH. Sodium reabsorption creates osmotic and electrical gradients, pulling water and chloride paracellularly, with water following sodium via osmosis and chloride tagging along due to charge attraction.
And we have these aquaporin water channels too.
Aquaporin water channels in the proximal tubule allow water to move transcellularly, driven by osmotic gradients created by sodium reabsorption via the sodium-potassium ATPase. These channels facilitate rapid water reabsorption alongside sodium, chloride, and bicarbonate, ensuring efficient fluid recovery from the tubular fluid.
Um, and glucose co-transported?
Yes, glucose is co-transported in the proximal tubule via sodium glucose co-transporters, SGLTs, where sodium's movement into the cell, driven by the sodium-potassium ATPase gradient, pulls glucose along. This transcellular reabsorption ensures nearly all filtered glucose is returned to the blood.
Okay, and so
All right.
tell us more about the glucose reabsorption. So, and we have proximal convoluted and proximal straight tubule. Explain us this.
Glucose reabsorption in the proximal tubule happens mainly in the proximal convoluted tubule, PCT, via sodium glucose cotransporters, SGLTs, where sodium's gradient, set by sodium potassium ATPase, drives glucose into cells, then into the blood, recovering nearly 100% of filtered glucose. The PCT handles most reabsorption due to its large surface area, while the proximal straight tubule, PST, continues limited glucose reabsorption but focuses more on water and urea, with fewer SGLT transporters.
What is this SGLT1 and 2 and apical membrane basolateral? Give us this terminology, explain it to us as simply as possible.
SGLT1 and SGLT2 are sodium glucose cotransporters on the apical membrane, facing the tubular fluid of proximal tubule cells, pulling glucose and sodium from the filtrate into the cell. Glucose exits through the basolateral membrane, facing the blood, via facilitated diffusion transporters, GLUTs, into the bloodstream, with SGLT2 handling most glucose and SGLT1 fine-tuning lower amounts.
So do we have fluoresceins as inhibitors of SGLT2? Dapagliflozin, empagliflozin.
Yes, flozins like dapagliflozin and empagliflozin, not hagliflozin, likely a mix-up, are SGLT2 inhibitors that block sodium glucose cotransporters in the proximal tubule, preventing glucose reabsorption and promoting its excretion in urine. This lowers blood sugar, making them effective for type 2 diabetes management.
Huh. And what other reabsorptions do we have in the PT? We have potassium, urea, amino acids. Tell us.
In the proximal tubule, PT, potassium is reabsorbed mainly paracellularly, following sodium and water gradients, while amino acids are reabsorbed transcellularly via sodium-coupled cotransporters, similar to glucose. Urea is reabsorbed passively, mostly paracellularly, driven by water reabsorption, with about 50% returning to the blood to maintain nitrogen balance.
And proteins is via receptor-mediated endocytosis. What is this?
Receptor-mediated endocytosis in the proximal tubule is when proteins in the tubular fluid bind to specific receptors on the apical membrane of tubule cells, triggering the cell to engulf the protein receptor complex into a vesicle. The vesicle is then processed inside the cell, breaking down proteins into amino acids for reabsorption into the blood, ensuring minimal protein loss in urine.
So, Grok, let me ask you an irrelevant question. Do you know the passport, the UK passport? Is it black or blue?
The new UK passport, issued since 2020, is officially navy blue, a return to the pre-EU design, though some say it looks almost black in certain lighting
It's
due
not
to its dark.
almost black, it's black.
Haha, all right, I hear ya. The UK passport is indeed listed as navy blue officially, but I get why you'd call it black. It can look super dark, especially under dim light.
Never mind
Wanna-
dim light, dude. It's not fucking blue, dude. Anyway, let's go back to studying. So when it comes to protein reabsorption, I'm still a bit confused. So in vesicles, they transport to the cell, and then what's going on?
Fair enough. We'll park the passport color debate. Back to protein reabsorption. In the proximal tubule, proteins in the tubular fluid bind to receptors on the cell's apical membrane, triggering receptor-mediated endocytosis, where the cell engulfs the protein receptor complex into a vesicle. Inside the cell, the vesicle fuses with lysosomes, which break down the proteins into amino acids that are then released across the basolateral membrane into the blood for reuse, preventing protein loss in urine.
Okay, and so
All
we
right,
have
you're
organic acids. Well, so we have two kinds of, sorry, we have organic acids and inorganic acids. Is there a difference between how they're secreted into the PT?
Organic acids, like drugs or metabolites, are secreted into the proximal tubule via specific transporters, e.g., organic anion transporters, on the apical membrane, actively pumped from blood to tubular fluid. Inorganic acids, like phosphate or sulfate, are typically reabsorbed rather than secreted, using sodium-coupled cotransporters, though excess may be excreted if blood levels are high.
What are para-aminohipurates?
Para-aminohipurate PAH, is an organic acid used to measure renal plasma flow because it's freely filtered by the glomerulus and actively secreted into the proximal tubule via organic anion transporters with minimal reabsorption. Its rapid clearance from blood into urine makes it a marker for assessing kidney function and blood flow through the nephrons.
Tell me again, I don't understand what this is.
Para-aminohyperate, PAH, is a synthetic organic acid given during medical tests to measure how much blood the kidneys filter, as it's completely filtered by the glomerulus and secreted into the proximal tubule without being reabsorbed. Its quick removal into urine helps doctors gauge renal plasma flow and kidney efficiency.
I'm confused about what it is. Can you? So, and it's not an endogenous compound.
Right. Para-aminohyperate, PAH, is not something your body naturally makes, not endogenous. It's a synthetic organic acid used in medical tests. It's given to measure kidney blood flow because it's filtered by the glomerulus and actively secreted into the proximal tubule, clearing from the blood into urine almost completely, showing how well the kidneys are working.
I see. So it's not filtered; it's only secreted. So we will see how good the kidneys secrete things.
Actually, para-aminohyperate, PAH, is both filtered by the glomerulus and secreted into the proximal tubule, which is why it's great for testing kidney function. Its near complete clearance, filtered plus secreted, with almost no reabsorption, measures renal plasma flow, reflecting both filtration and secretion efficiency.
Are you sure? I can see here it's only about secretion.
My apologies if I caused confusion. You're right to question. Let's clarify. Para-aminohyperate, PAH, is indeed both filtered by the glomerulus and actively secreted into the proximal tubule via organic anion transporters. But its secretion is the dominant process for its clearance, which is why it's often emphasized in tests of tubular secretion. The combination of filtration and near complete secretion with minimal reabsorption allows PAH to measure effective renal plasma flow, as almost all PAH entering the kidney is excreted in urine. Want me to dig into a specific aspect or source to align with what you're seeing?
That's all good. So, and how does alpha ketoglutarate and dietary carboxylates connect?
Alpha ketoglutarate, an organic acid in the Krebs cycle, is exchanged in the proximal tubule for dietary carboxylates, like citrate or lactate, during their secretion via organic anion transporters. This exchange helps secrete dietary carboxylates.
Yeah.
All right, let's make sure we're on the same page. Since you're seeing something suggesting PHH is only about secretion, can you share a bit more about what you're looking at?
No,
Or I can.
okay, never mind that. Let's focus on the alpha ketoglutarate and dietary carboxylates.
All right, let's shift gears to alpha fetoprotein, AFP, and di and tricarboxylates. Unfortunately, there's no.
Alpha ketoglutarate, I said, and how it connects with the para-aminohyperate.
My bad for the mix-up. Alpha ketoglutarate, a dicarboxylate, is key in the secretion of para-aminohyperate, PHH, in the proximal tubule. It acts as a counterion in organic anion transporters, OATs, where alpha ketoglutarate is exchanged from inside the cell for PHH from the blood, driving PHH secretion into the tubular fluid.
All right, and now let's talk a bit about endogenous acids and bases and some drugs that have to do with all of this.
Endogenous acids, like uric acid, and bases, like ammonia, are secreted in the proximal tubule via organic anion. OAT, and cation, OCT, transporters, respectively, to regulate pH and clear metabolites. Drugs like probenecid inhibit OATs, reducing secretion of acids like PHH or penicillin, while others like cimetidine block OCTs, affecting base secretion.
What about acetazolamide and chlorothiazide?
Haha, ich glaube, du meinst vielleicht
Dude, I was
Acetylsalicylate.
talking to you in English. I asked you about acetazolamide and chlorothiazide.
My bad, got thrown off by the German. Acetazolamide and chlorothiazide are drugs affecting the proximal tubule. Acetazolamide inhibits carbonic anhydrase, reducing bicarbonate reabsorption to promote diuresis and manage pH, while chlorothiazide blocks sodium chloride reabsorption, increasing urine output. Want more on their mechanisms or effects?
Well, I wonder what in their name gives it away that they have to do with the PT.
The names acetazolamide and chlorothiazide don't directly signal proximal tubule PT action, but their suffixes and class hint at their function. Zolamide in acetazolamide links to carbonic anhydrase inhibitors, which act in the PT to block bicarbonate reabsorption. And thiazide in chlorothiazide indicates a thiazide diuretic, primarily affecting the distal tubule, but with some PT sodium chloride reabsorption effects. Drug names often reflect
Never
chemical
mind,
structure.
alright. Okay, let's start talking a bit about the loop of Henle now. So, um,
The loop of Henle has two main parts, the descending limb and the ascending limb. In the descending limb, water is extracted from the filtrite into the surrounding interstitial fluid due to high osmotic pressure, concentrating the urine. In the ascending limb, sodium and chloride ions are actively reabsorbed into the interstitial fluid, but water is not, diluting the filtrite.
but why is water in the descending and then sodium and chloride in the ascending?
Water is reabsorbed in the descending limb because it's permeable to water in the surrounding medullary interstitium, has a high osmotic gradient, pulling water out to concentrate the filtrite. The ascending limb is impermeable to water, but actively reabsorbs sodium and chloride via transport proteins, creating that osmotic gradient in the medulla while diluting the filtrite. This countercurrent mechanism maximizes urine concentration and conserves water.
I don't understand the reasoning.
All right, let's break it down simply. The descending limb is like a sponge for water. It's permeable, so water gets pulled out into the salty, concentrated medullary interstitium, making the urine more concentrated. The ascending limb, on the other hand, is watertight, but pumps out sodium and chloride ions
Yeah,
using
but
energy.
why not the other way around? Why not throw the sodium first?
Good question. The loop of Henle's set up water reabsorption in the descending limb and sodium chloride in the ascending is designed to create a strong osmotic gradient in the kidney's medulla for efficient water conservation. If sodium and chloride were reabsorbed first in the descending limb, the filtrite would lose salt early, making it less concentrated.
So
and
why
weakening
are
the
the
osmotic
cells
pull need-
why are the cells in the descending limb relatively flat and not very sophisticated and developed? Huh?
The cells in the descending limb are flat and simple, like squamous epithelial cells, because their main job is passive water reabsorption, driven by the osmotic gradient in the medulla, not active transport. They don't need complex structures or lots of mitochondria, since they're not using energy to pump ions. Unlike the thicker, mitochondria-rich cells in the ascending limb that actively reabsorb sodium and chloride, This structural difference matches their distinct roles in the loop's countercurrent
I
system.
see. And why is it? And so the thickness is also because of this. Like we have more thickness in the ascending because we have more jobs for it.
Exactly. The ascending limb has thicker, more complex cells, cuboidal or columnar, with lots of mitochondria, because they're doing heavy lifting, actively pumping sodium and chloride out using energy. The descending limb's flat, simple cells are just letting water pass passively, so they don't need the extra thickness or energy machinery. It's all about matching structure to function.
Hmm. Okay. So, I see. So the tubular wall, if it's thick, it's impermeable to water. If you could summarize in a sentence why it's water first and not water second, what would you say?
The descending limb reabsorbs water first because its thin, permeable wall allows
Oh no,
passive
this
water.
is not a good argument because then I will ask you why is it thin, and then you're gonna say it's thin so it can get water. So instead of getting stuck into this loophole, let's find a better answer, shall we?
Fair point. Let's avoid the circular trap. The loop of Henle reabsorbs water first in the descending limb to concentrate the filtrite early, leveraging the medulla's osmotic gradient, while the ascending limb's active sodium and chloride reabsorption builds that gradient without water loss, ensuring efficient urine concentration. This sequence maximizes water conservation by creating and using the medullary gradient in a countercurrent flow.
Okay, good. And I don't understand the answer. What exactly are we taking advantage of by doing it first?
By reabsorbing water first in the descending limb, we're taking advantage of the high salt concentration already present in the kidney's medulla, which pulls water out passively to concentrate the filtrate. This sets up a concentrated filtrite that the ascending limb can then dilute by pumping out
We'll
sodium and
come back to
chloride.
it. We'll come back to it because I'm a bit confused. So counter current multiplication. What is this?
All right, no worries. We'll circle back. Countercurrent multiplication in the loop of Henle is the process where the descending and ascending limbs work together to amplify the osmotic gradient in the kidney's medulla. The descending limb concentrates the filtrite by losing water, while the ascending limb pumps out sodium and chloride, making the medulla saltier and enabling the kidney to produce concentrated urine efficiently.
So it's basically what we are talking about.
Yeah, exactly. Countercurrent multiplication is just the fancy term for how the descending and ascending limbs team up to
Yeah,
create.
got it. And why do we go from cortex to outer medulla to inner medulla?
All right, so the loop of Henle's path from cortex to outer medulla to inner medulla is key to building a stronger osmotic gradient deep in the kidney. It starts in the cortex, where the filtrite is still dilute, dips into the outer medulla, where water begins to exit in the descending limb, and reaches the inner medulla, the saltiest region, where the gradient peaks. This
And why
progression let
why do we call it outer and inner medulla?
the outer and inner medulla are named based on their anatomical positions in the kidney. The outer medulla is closer to the cortex, where the osmotic gradient starts building, while the inner medulla is deeper, where the gradient gets super concentrated due to the loop of Henle's countercurrent multiplication. It's just a way to describe the kidney's layered structure as you move toward its core.
I don't get. Okay, I see. I see. That makes sense. And then we go to the distal tubule.
Yep, exactly. After the loop of Henle, the filtrite flows into the distal tubule, which is back in the cortex. There, It fine-tunes the reabsorption of sodium, potassium, and water, and adjusts pH based on the body's needs before heading to the collecting duct.
And so we have renal outer medullary potassium channels.
Yeah, you're talking about the Rampke channels, renal outer medullary potassium channels. These are in the ascending limb and distal tubule, where they help secrete potassium into the filtrite to maintain potassium balance and support the sodium-potassium pump's activity. They're key for keeping the electrical gradient that drives ion reabsorption.
Okay. Whereas before it wasn't a sodium chloride co-transporter.
Right, you're connecting some dots here. In the thick ascending limb of the loop of Henle, the sodium potassium chloride co-transporter, NKCC2, is the main player, pulling sodium,
Excuse me, I
potassium.
thought we were talking about the distal tubule, not the thick ascending limb.
My bad, thanks for catching that. In the distal tubule, you're right. It's not the sodium potassium chloride co-transporter, like in the thick ascending limb. Instead, the distal tubule uses the sodium chloride co-transporter, NCC, to reabsorb sodium and chloride together, fine-tuning electrolyte balance, while Rampke channels handle potassium secretion.
So why this difference?
The difference comes down to the distinct roles of the thick ascending limb and distal tubule in the nephron. The thick ascending limb uses the sodium potassium chloride co-transporter, NKCC2, to powerfully reabsorb sodium, potassium, and chloride, creating a strong osmotic gradient in the medulla for urine concentration. The distal tubule, however, uses the sodium chloride co-transporter, NCC, for more precise, regulated reabsorption of sodium and chloride, adjusting electrolyte and fluid balance based on the body's needs.
Yeah,
With
but I
Rampke-
don't get why we have NAK TPAs or whatever, and then this we have it in the first stage in the proximal, and then in the loop of Henle we have this sodium potassium whatever it's called, and now we have another one. Explain to me in 3 sentences for each case why that one is the best one.
All right, let's clarify why each nephron segment uses its specific transporters. Sodium potassium ATPase in the proximal tubule, sodium potassium chloride co-transporter, NKCC2, in the thick ascending limb, and sodium chloride co-transporter, NCC, in the distal tubule, in 3 sentences each. Proximal
No,
tubule.
1 sentence speech.
The proximal tubule uses sodium potassium ATPase to drive reabsorption of most sodium, glucose, and amino acids, setting up bulk filtrite processing. The thick ascending limb employs the sodium potassium chloride co-transporter, NKCC2, to reabsorb sodium, potassium, and chloride, creating a strong medullary osmotic gradient for urine concentration. The distal tubule uses the sodium chloride co-transporter, NCC, to finely adjust sodium and chloride reabsorption, balancing electrolytes based on hormonal signals.
Yeah, I don't get the reason that each one has that one though.
All right, let's dive into why each transporter fits its specific nephron segment. The sodium potassium ATPase in the proximal tubule is there because it powers the massive reabsorption of about 70% of sodium, water, glucose, and other
Yeah,
solutes.
but what I want is why those are better than the other ones in each option.
Got it. You want to know why each transporter is the best fit for its nephron segment compared to the others. The sodium potassium ATPase is ideal for the proximal tubule because it provides a strong electrical gradient to drive co-transport of sodium with glucose, amino acids, and other solutes, handling bulk reabsorption efficiently, unlike the more specialized NKCC2 or NCC. The sodium potassium chloride cotransporter, NKCC2, is perfect for the thick ascending limb because it reabsorbs sodium, potassium, and chloride together in a 1:1:2 ratio, maximizing the medullary osmotic gradient for urine concentration, which the ATPase alone, or NCC, couldn't achieve as effectively. The sodium chloride cotransporter, NCC, suits the distal tubule because it allows precise hormone-regulated reabsorption of sodium and chloride without potassium, fine-tuning electrolyte balance, unlike the high-capacity NKCC2 or the broader ATPase-driven system.
I'll be honest, dude. I still haven't understood why each one. Okay, each one is ideal for that stage.
No worries.
Actually, let's put a break to this and let's talk about the sodium. So it's exchanged for potassium in the distal tubule in and in the late stage, and I wonder why and what do principal cells have to do with this? Why they are called principal?
All right, let's switch gears and talk about sodium-potassium exchange in the late distal tubule and collecting duct, and the role of principal cells. In the late distal tubule and collecting duct, sodium is reabsorbed in exchange for potassium secretion, driven by the sodium-potassium ATPase and aldosterone-regulated epithelial sodium channels, ENAC, with potassium exiting via Romke channels to balance electrolytes. Principal cells handle this exchange because they're the main cell type in these segments, called principal due to their abundance and primary role in sodium reabsorption and potassium secretion, unlike intercalated cells that focus on acid-base balance.
So, wait a sec. So we have this exchange, but I still don't understand why do we have it late in the tubule, and I don't understand why the principal cells are called as such.
All right, let's clarify those 2 points simply. The sodium-potassium exchange happens late in the distal tubule and collecting duct because this is where the body fine-tunes electrolyte balance, using aldosterone to control sodium reabsorption and potassium secretion based on what the body needs, unlike earlier segments focused on bulk reabsorption. Principal cells are called that because they're the most abundant cells in these segments and do the main job of handling sodium and potassium exchange, distinct from the less common intercalated cells that deal with pH regulation.
And they are sensitive to aldosterone. Also, tell me about the ROMK channel and how potassium relates to it.
Yeah, principal cells are sensitive to aldosterone, which boosts sodium reabsorption and potassium secretion by upregulating epithelial sodium channels, ENAC, and sodium-potassium ATPase. The Romke, renal outer medullary potassium, channels in principal cells allow potassium to be secreted into the filtrite, driven by the ecoelectrical gradient created when sodium is reabsorbed. This potassium secretion is crucial for maintaining blood potassium levels, especially when aldosterone signals the kidneys to conserve sodium and excrete excess potassium.
Tell me more.
All right, let's dive deeper into the principal cells, aldosterone, and Romke channels in the late distal tubule and collecting duct. Aldosterone, a hormone released when blood sodium is low or potassium is high, binds to receptors in principal cells, ramping up the expression of ENAC channels and sodium-potassium ATPase to reabsorb sodium and create a favorable gradient for potassium to exit through Romke channels into the urine. Romke channels are potassium-selective pores that stay open to secrete potassium efficiently, especially under aldosterone's influence, ensuring the body dumps excess potassium while conserving sodium for blood pressure and fluid balance. This tightly regulated process in principal cells is critical for electrolyte homeostasis, and any disruption, like in certain kidney diseases, can mess up potassium or sodium levels big time.
Okay, tell me more about this ROMK channel thing.
All right, let's zoom in on the ROM-key channels. ROM-key, or renal outer medullary potassium channels, are specialized potassium channels found in the principal cells of the late distal tubule and collecting duct, where they play a key role in secreting potassium into the urine to regulate blood potassium levels. They work by allowing potassium to flow out of the cell into the filtrite, driven by the ecoelectrical gradient set up when sodium is reabsorbed through ENAC channels, and the sodium potassium ATPases pump sodium out and potassium into the cell, with aldosterone boosting this process to enhance potassium secretion when needed.
Okay. And
So,
let
to
me see.
continue, ROM key channels are crucial for potassium homeostasis because their activity is finely tuned by factors like aldosterone, pH, and even certain kinases that can adjust how open or closed they are. They're structured to be highly selective for potassium, ensuring efficient secretion without letting other ions mess things up. And mutations in ROM key can lead to disorders like Barter's syndrome, where potassium and sodium balance goes haywire. Anything specific about ROM key or the principal cells you want to dig into next?
So the overall effect we have with aldosterone is we have more sodium reabsorbed, so more water moves from the tubular fluid back into the plasma, so blood pressure increases. Could you explain this a bit more?
Absolutely. Let's break down how aldosterone's effects tie together. Aldosterone acts on principal cells in the late distal tubule and collecting duct, increasing the number of ENAC channels and sodium potassium ATPase pumps, which boosts sodium reabsorption from the tubular fluid into the blood. This sodium movement creates an osmotic gradient that pulls water back into the plasma through aquaporin channels, regulated by vasopressin, increasing blood volume and consequently raising blood pressure to maintain proper circulation and fluid balance.
Okay, let's take a 1 minute break. You guys might find this interesting too. I want to ask, can the Whoop band let me know when my heart rate goes over 100? Can I set it up so there's a notification coming?
All right, cool. Let's switch to the Whoop band for a sec. The Whoop band itself doesn't directly send real time notifications for heart rate exceeding a threshold like 100 BPM, as it's designed more for tracking and analyzing data post activity rather than live alerts. However, you can view your heart rate trends in the Whoop app, and if you're using it with a connected device or app like Strava or Zwift that supports live heart rate broadcasting via Bluetooth, you might set up alerts through those platforms, depending on their features.
Is there anything I can wear on me that can do this though? That can kind of let me know when my heart rate, it can buzz maybe when my heart rate goes over 100. I read in a book, I think it's in a book, How to Have a Healthy Marriage, something like that, that it said that there's something to wear that's like that.
Yeah, there are wearable devices that can buzz or notify you when your heart rate goes over 100 BPM. And it sounds like you're looking for something like what's mentioned in that book, possibly for stress or emotional monitoring. Devices like the Apple Watch, Fitbit, e.g., Versa or Charge models, Garmin smartwatches, and
So
Polar fitness
Apple Watch
trackers.
can do it. Tell me how Apple Watch can do it.
The Apple Watch can notify you when your heart rate exceeds 100 BPM by using its built in optical heart rate sensor and customizable settings in the heart rate app. To set it up, open the Apple Watch app on your iPhone, go to My Watch, tap Heart, then select High Heart Rate and choose a threshold like 100 BPM. The watch will vibrate and alert you if your heart rate exceeds this while you're inactive for 10 minutes. Note that this feature won't trigger during detected workouts, and low power mode may disable it.
Hmm. So let me continue now. I want to ask you a bit more about the renal actions of aldosterone. So, how do drugs such as spironolactone and eplerenone can be used to block the aldosterone action, and how exactly do they block it?
All right, let's zoom in on aldosterone and its renal actions, plus how drugs like spironolactone and eplerenone throw a wrench in the works.
Yeah, and let's
Aldosterone
let's do it in two, three sentences.
Aldosterone boosts sodium reabsorption and potassium excretion in the kidney's distal tubule and collecting duct by binding to mineralocorticoid receptors, triggering gene expression for ion channels and pumps. Spironolactone and eplerenone are mineralocorticoid receptor antagonists that competitively bind these receptors, blocking aldosterone's effects and reducing sodium reabsorption and potassium loss. This helps manage conditions like hypertension and heart failure by countering fluid retention.
Yeah, but how exactly do they do it? Spironolactone and eplerenone, and how exactly does the name relate to the function and the structure relates to the function?
All right, let's get precise about how spironolactone and eplerenone block aldosterone's action and how their names and structures tie into their function. Aldosterone normally binds to mineralocorticoid receptors in the cells of the distal tubule and collecting duct, activating them to increase sodium reabsorption and potassium excretion through gene expression that ramps up ion channels and pumps, like the epithelial sodium channel ENAC. Spironolactone and eplerenone are competitive antagonists, meaning they bind to the same mineralocorticoid receptor site as aldosterone but don't activate it, effectively blocking aldosterone from docking and triggering its effects. This reduces sodium reabsorption and potassium loss, which is why they're used for conditions like heart failure or hypertension to curb
Okay.
fluid.
So the intercalated cells, we talked about principal cells. Now I want us to talk a bit about intercalated cells. Why are they the ones that act on the collecting duct instead of principal cells? What's the difference?
All right, let's shift gears to intercalated cells in the collecting duct and why they're distinct from principal cells. Principal cells in the collecting duct handle sodium reabsorption and potassium secretion, largely driven by aldosterone, using channels like ENAC and ROMK. Intercalated cells, on the other hand, are specialized for acid-base balance. They come in two types. Alpha intercalated cells secrete hydrogen ions to acidify urine and reabsorb bicarbonate, while beta intercalated cells do the opposite, secreting bicarbonate to alkalinize urine. This happens via proton pumps, H plus ATPs, and chloride bicarbonate exchangers, not the sodium potassium mechanisms of principal cells. Intercalated cells take on this role because their molecular machinery is tailored for pH regulation, whereas principal cells are wired for electrolyte balance.
And
The collecting
there's also hydrogen sodium and hydrogen potassium exchange now instead. So you have the sodium hydrogen ATPase. And I wonder why the distal tubule is more ideal place for this type of transporter instead of the previous one we saw in the beginning, NAK ATPase. And explain to me the differences between alpha and beta intercalated cells. Do it in four sentences.
The distal tubule, including the collecting duct, is ideal for sodium hydrogen NHE and hydrogen potassium exchangers because it fine tunes acid base balance and electrolyte homeostasis in response to systemic needs. Unlike the NAK ATP in the proximal tubule, which primarily drives bulk reabsorption, alpha intercalated cells in the collecting duct secrete hydrogen ions via H plus ATPs and H plus K plus ATPs to acidify urine and reabsorb bicarbonate, helping correct acidosis. Beta intercalated cells, conversely, secrete bicarbonate and reabsorb hydrogen ions through chloride bicarbonate exchangers, aiding in alkalosis correction. These distinct roles of alpha and beta cells allow precise pH regulation, complementing the principal cell's focus on sodium and potassium balance.
Hmm. didn't understand much of that. Maybe if you summarize it in a sentence or two.
The distal tubule uses sodium hydrogen and hydrogen potassium exchangers to regulate acid-base balance, unlike the NAK ATPs in the proximal tubule, which focuses on reabsorption. Alpha intercalated cells acidify urine to fix acidosis, while beta intercalated cells release bicarbonate to correct alkalosis.
Well, okay, and so so this is kind of acid-base regulation.
Yeah, exactly. This is all about acid-base regulation in the kidneys. The intercalated cells in the distal tubule and collecting duct are key players in keeping your blood pH balanced by tweaking urine acidity or alkalinity. Want me to dive deeper into how this ties into the bigger picture of pH control or something specific about these cells?
Um, well, I wanna just a minute. I just lost my notes here. Just give me a sec. Hmm. No, I'm good. I want to talk a bit about the collecting duct, which is with the final step. So this is kind of relatively impermeable to the movement of things. But we have ADH. Can you talk to us about ADH and the collecting duct in two or three sentences?
The collecting duct is typically impermeable to water, but antidiuretic hormone, ADH, also called vasopressin, makes it permeable by triggering the insertion of aquaporin 2 water channels into the cell membranes. This allows water reabsorption, concentrating the urine and helping regulate body fluid balance. ADH's action is crucial for responding to dehydration or high blood osmolarity, ensuring water is conserved.
Ah, and so ADH, does it have to do with sodium or only with water?
ADH, or vasopressin, primarily deals with water, not sodium. It acts on the collecting duct to increase water reabsorption by inserting aquaporin 2 channels, which lets water move back into the bloodstream, concentrating the urine. While this can indirectly affect sodium concentration in the blood by altering water volume, ADH doesn't directly regulate sodium levels. That's more the job of aldosterone and principal cells.
So what does it do to CAMP and to the aquaporin channels? And what's going on with why is it called a non-peptide? And what is it? Yeah, what's its MW? And tell me
All right.
about the V2 receptors too. Maybe in total everything I just asked, four sentences.
ADH, a nonapeptide named for its 9 amino acid structure, with a molecular weight of about 1084 day, binds to V2 receptors in the collecting duct, activating a CAMP signaling pathway that prompts aquaporin 2 channels to insert into cell membranes, increasing water reabsorption. V2 receptors, found mainly in renal collecting duct cells, are G protein coupled receptors that trigger adenylate lysis to produce CAMP upon ADH binding, amplifying the water permeability response. The term nonapeptide reflects ADH's peptide chain length, distinguishing it from other hormones. This mechanism ensures efficient water conservation to maintain body fluid balance.
Interesting. Let me ask you about urea. So we have it's also reabsorbed from the collecting duct, right? And it kind of helps with creating a hypertonic environment. If you could talk a bit about this.
Yeah, urea plays a fascinating role in the collecting duct. It's indeed reabsorbed there, especially in the medullary collecting duct, and this helps create a hypertonic environment in the kidney medulla, which is crucial for concentrating urine. Here's how it works. Urea is filtered by the glomerulus and partially reabsorbed in earlier nephron segments, but in the collecting duct, specific urea transporters like UTA1, facilitated by ADH, allow urea to move out of the duct into the medullary interstitium. This boosts the medullary osmotic gradient, drawing water out of the collecting duct, also thanks to ADH's aquaporin 2 channels, which concentrates the urine. Without urea's contribution to this hypertonic environment, The kidney's ability to conserve water and produce concentrated urine would be way less effective. Want me to dig into any specific part of this process or its regulation?
I'm a bit confused. Also, can I ask if you could explain this again in one or two sentences to summarize? And also, I wonder what happens to all the solutes and the water that's reabsorbed from the tubule?
Urea is reabsorbed in the collecting duct via urea transporters, enhancing the medullary osmotic gradient to help concentrate urine by drawing water out. Solutes like sodium, potassium, and urea, along with water reabsorbed from the tubule, mostly return to the bloodstream via peritubular capillaries, maintaining body fluid and electrolyte balance, while excess solutes and minimal water are excreted as urine.
And they go to the peritubular capillaries and the vasa recta. What are these?
The peritubular capillaries in vasa recta are specialized blood vessels in the kidneys that play a key role in handling reabsorbed solutes and water. Peritubular capillaries surround the proximal and distal tubules in the cortex, picking up water, sodium, glucose, and other solutes
But why are
reabsorbed.
they called the way they are called?
The names peritubular capillaries and vasa recta reflect their location and structure in the kidneys. Peritubular capillaries are called so because they surround the tubules, like the proximal and distal convoluted tubules, forming a dense network in the cortex to collect reabsorbed water and solutes. Vasa recta, meaning straight vessels in Latin, refers to the straight, hairpin-shaped capillaries that run parallel to the loops of Henle in the medulla, helping maintain the medullary osmotic gradient by exchanging water and solutes. These names directly describe
Why
their
is
anatomy.
the ADH's ideal place to be synthesized the hypothalamus, and why is it released ideally from the? Why is the ideal place to be released the posterior pituitary? If you could answer in a sentence and a half.
ADH is ideally synthesized in the hypothalamus because its neurons can detect blood osmolarity changes, triggering production of this nonapeptide. It's released from the posterior pituitary, an extension of the hypothalamus, which serves as an efficient storage and release site directly into the
Yeah, but
bloodstream.
why from the posterior, not the anterior?
The posterior pituitary is ideal for ADH release because it's essentially an extension of the hypothalamus, where ADH is synthesized, and its axons store and secrete ADH directly into the bloodstream for rapid action.
And
The
how?
anterior
Why does nicotine and morphine and ether and barbiturates? Why do they increase ADH release?
nicotine, morphine, ether, and barbiturates can increase ADH release by stimulating the hypothalamus or posterior pituitary, often through indirect effects on the central nervous system. These substances can mimic or amplify stress signals, trigger pain pathways, or alter neural activity, which the hypothalamus interprets as a need for water retention, prompting ADH secretion from the posterior pituitary to reduce urine output. For example, nicotine stimulates nicotinic receptors, enhancing sympathetic activity that can signal the hypothalamus, while morphine and barbiturates may depress certain neural pathways, indirectly boosting ADH release. The exact mechanisms vary, but they all converge on activating the neurocrine reflex that controls ADH. Want me to focus on any specific drug's effect or
And I
related?
want to ask about diabetes in speed in insipidus and how this is caused by a lack of ADH.
Diabetes insipidus is caused by a lack of ADH, or the kidney's inability to respond to it, leading to excessive water loss. Without enough ADH, the collecting duct remains impermeable to water, so the kidneys can't reabsorb it, resulting in large amounts of dilute urine and intense thirst. This can stem from hypothalamic damage, reducing ADH production, central diabetes insipidus, or from defective kidney receptors for ADH, nephrogenic diabetes insipidus. Want me to explain the differences between these types or how they're treated?
Hmm. Yeah, and what does insipidus mean?
Diabetes insipidus is a condition where a lack of ADH, or the kidney's inability to respond to it, causes excessive dilute urine and thirst due to poor water reabsorption in the collecting duct. The term insipidus comes from Latin meaning tasteless, referring to the urine's lack of flavor compared to the sweet, glucose-rich urine in diabetes mellitus. Central diabetes insipidus results from hypothalamic or pituitary damage,
And this is
reducing
because of the difference in water too. Basically, not only glucose.
Exactly. The key difference in diabetes insipidus is the water issue, not glucose. Unlike diabetes mellitus, where high blood glucose causes sweet, concentrated urine, diabetes insipidus results from a lack of ADH or kidney response to it, leading to excessive water loss and dilute, tasteless urine. This happens because the collecting duct can't reabsorb water without ADH's action on aquaporin 2 channels. So water just passes through, regardless of glucose levels. Want to
So
dive
in
into how
insipidus we have extra water or no water?
in diabetes insipidus, you lose extra water, not retain it. The lack of ADH, or the kidney's inability to respond to it, means the collecting duct can't reabsorb water, so you excrete large amounts of dilute urine, leading to dehydration and intense thirst as your body tries to compensate for the water loss. Want me to clarify how this affects the body further or something else about it?
This a lot of water makes the pee less sweet.
Yeah, exactly. In
Okay, that
diabetes
makes sense. And let us close back with the urea just to make sure. Why is the hyper environment becoming hypertonic? And explain to us how it enters the interstitium and then how it increases osmolarity and why this promotes water reabsorption.
All right, let's wrap up with urea and the hypertonic environment in the kidney. Urea helps create a hypertonic medullary interstitium because it's reabsorbed from the collecting duct, especially in the inner medulla, through urea transporters like UTA1, which are activated by ADH. This reabsorbed urea accumulates in the interstitium, increasing its osmolarity, often to 1200 mosm per liter or more, making it much saltier than the surrounding fluid. The high osmolarity pulls water out of the collecting duct via aquaporin 2 channels, also ADH dependent, promoting water reabsorption to concentrate the urine and conserve body water. This whole setup is why the medullary gradient is so crucial for the kidney's ability to produce concentrated urine when you're dehydrated. Want me to tie this back to anything else or clarify a specific part?
Are we confused? Yeah, explain it again, summarize it in a sentence or two.
Urea is reabsorbed from the collecting duct into the medullary interstitium via ADH-activated transporters, creating a hypertonic environment that increases osmolarity and pulls water out of the duct to concentrate urine. This high osmolarity in the medulla enhances water reabsorption, helping conserve body water.
So it increases the reabsorption. I don't get it.
Okay, let's break it down simply. Urea gets reabsorbed from the collecting duct into the kidney's medullary interstitium, making that area super concentrated, or hypertonic, because it raises the osmolarity. This high concentration acts like a magnet, pulling water out of the collecting duct through aquaporin 2 channels, thanks to ADH. So more water is reabsorbed into the bloodstream instead of being lost.
So it lives with urea.
Exactly. You nailed
Got
it.
it. All right,
The water gets
guys. Thanks for watching, and I'll see you next time.